Assalamu alaikum. Good afternoon. Welcome to 5th International Conference on Applied Zoology 2022. I am Hafsa Maham, MPhil scholar from Government College University, Faisalabad. I am going to moderate this session on the behalf of Applied Zoological Society of Pakistan. This session includes different talks from drug discovery, physiology, epidemiology, cancer biology, and entomology. Our first presenter of this session is Dr. Mehwi Salman from Government College University, Faisalabad. She is going to present her topic, Bioprotection of Z-Maze L from aflatoxigenic aspergillus flavus by Legolactobacillus coriniformis BCH4. Please, Dr. Mehwi Salman.
Hello, everyone. First of all, I would like to thank the organizers of this conference for providing me an opportunity to present my research work in 5th International Conference on Applied Zoology. The title of my presentation is The Bioprotection of Z-Maze from Aflatoxigenic Aspergillus flavus by Legolactobacillus coriniformis BCH4. These are the outlines of my presentation. First of all, let me introduce the background of my research. The Z-maize is one of the most important cereal crop. It is also known as corn or maize and is most cultivated crop in the world. It is not only used as food, but also for animal feed and as raw material for the production of biofuels. It is the highest yielding among world's major crops due to its low cultivation cost. The Z-maize, it faces different infections, especially the fungal infection due to the presence of different fungal genera like Fusarium, Penicillium, Rhizopus, Aspergillus, and Titania during its germination, growth, and storage. Moreover, these pathogens also cause maize rot, kernel discoloration, loss of viability, mycotoxin contamination, and subsequent seedling blights. Deterrence of food from pathogenic microorganisms has always been a major task. The harmful products of such microorganisms is a major cause of risk for human as well as animal health. To overcome these risks, lactic acid bacteria have been used as bioprotective from few decades because of their safe nature. These labs produce various metabolites, including organic acids like lactic acid, acetic acid, and the phenyl lactate, bacteriocins antifungal peptides, luteins diacetyl fatty acids, and cyclic dipeptides, etc. These all secondary metabolites cumulatively influenced against fungal species as bioprotecting metabolites. The main objectives of my research is the in vitro biotic stress tolerance potential of Legolactobacillus coriniformis BCH4 against Aspergillus flavus. Moreover, we also employed this effect for the bioprotection of Z-maize against A flavus. First of all, the methodology, we concluded the in vitro biotic stress tolerance of Legolactobacillus coriniformis, and after evaluating this effect this potential, we employed this biotic stress tolerance against aflatoxigenic Aspergillus flavus. After concluding the biotic stress tolerance, we do fermentation production of antimicrobial compounds by using fermenter, and after fermentation production or after the production of metabolites, we do centrifugation for the collection of cell-free supernatant, and which was further filtered through 0.45 micron filters, and which was the filtrate was lyophilized and employed for bioprotective effect on maize grains. After that, the different treatments of maize grains were employed for FDIR to evaluate the functional groups of nutritional contents of maize grains, and also for the estimation of total phenolic flavonoid contents and antioxidant potential of maize grains, which was also followed by the determination of aflatoxins AFB1 and AFB2 by using HPLC. HPLC was also used for the detection of various phenolic acids in L-coroniformis BCH4 CFS. Now we come to the results. This is the biotic stress tolerance potential of Loigolactobacillus coroniformis, and we can find out clear determination of zone of L-coroniformis BCH4 CFS against Aspergillus flavus. That is the bioprotective effect of cell-free supernatant of Loigolactobacillus coroniformis BCH4 against A flavus. These are the two treatments. One is B1 is the treatment of our maize grains with CFS of L. coroniformis, and A1 is the control, that is the treatment of maize grains only with MRS broth without any culture of L. coroniformis. This is the FTIR spectra of maize grains. First of all, I would like to tell that three treatments, that is the T1, T2, and T3. T1 is the raw maize grains. T2 is the MRS broth with A flavors that is used as control against our treatment. And T3 is the treatment of maize grains with A flavors plus the CFS of L. coroniformis, that is our targeted bacteria. And we found that the FTIR spectrum of T3 has an improved outcome of different stretchings that we can visualize here with a reddish brown shade. The stretchings of carbonyl, CH, OH, and NH improved in T3 treatment. Similarly, these are the phenolic flavonoid contents and antioxidant potential, and we can also visualize from these graphs. A represents the graphical representation of three treatments of phenolics. B is for flavonoids, and C is for antioxidant activity percentage. And here T3 has improved results in both in phenolics, flavonoids, and antioxidants. These are this one is the aflatoxins production determination, that is the AFB1 and AFB2 in different treatments. This one is a chromatogram of T1, that is the raw without any treatment, which we can compare with T2, that is the MRS broth plus A flavors treated maize grains that we used as control in comparison with our T3 treatment. We can visualize here the presence of AFB1 and AFB2, a considerable concentration of both these aflatoxins. When we compare this T2 with T3, we didn't found any considerable concentration of this B1 and B2 in our T3 treatment. So it was concluded that our collected CFS of L. coroniformis BCH4 has a strong anti-aflatoxigenic effect against A flavors. This last one is the detection of phenolic acids. We also concluded by our research that these resultant phenolic acids also participate as an participated in antifungal potential of CFS of L. coroniformis, and we found different phenolic acids like chlorogenic acid, p-cumaric acid, 4-hydroxybenzoic acid, cafeteric, synapic, salicylic acid, and benzoic acid. This is the resultant chromatogram of phenolic acid detection by HPLC. Now I would like to conclude my research work. The present work represented the Loigo-Lactobacillus coroniformis PCH4 could be a good biotic stress tolerant bacterium. It was observed that L. coroniformis CFS has strong NB-aflatoxigenic potential against A. flavus-on-maize grains. The current findings also highlight higher nutritional total phenolic, flavonoid, and antioxidant contents in CFS treated maize grains in comparison with raw or A flavors infected grains. Moreover, this resultant biocontrol agent could also be used to improve the nutritional values and shelf life of other cereal grains that we will inshallah do in the future. The detailed characterization of this bacterium will be helpful as biocontrol agent and to explore its other beneficial effects in the future that we are also working on these aspects. And thank you so much for your attention. Thank you.
Thank you, Dr. Mehwish Salman, for productive talk. Our next presenter is Dr. Azhar Rafiq from Government College University, Faisalabad. Her topic of presentation is prevalence and intensity of gastrointestinal nematodes infections among wild sheep kept in captivity in Faisalabad, Pakistan. Please, Dr. Azhar Rafiq.
Bismillahirrahmanirrahim. I am very thankful to 5th International Conference on Applied Zoology 2022, which is organized by the Applied Zoological Society of Pakistan and Khwaja Farida University of Engineering and Information Technology, Rahim Yar Khan, Punjab, Pakistan. I am Dr. Azhar Rafiq, Assistant Professor, Department of Zoology, Government College University, Faisalabad. The topic of my presentation is prevalence and intensity of gastrointestinal nematodes infection among wild sheep kept in captivity in Faisalabad, Pakistan. First of all, introduction. Helminths are parasitic worms that were feed on living organism, living host to gain nourishment and safety while inflicting terrible nourishment absorption, weakness, and disorder within the host. The foremost gastrointestinal nematodes infecting the small ruminants include Haemonchus contortus, Trichostrongylus species, etc. Parasitic gastroenteritis infestation generally cause profuse diarrhea, decreased performance, weight loss, and in some cases, anemia. Wild sheep are very sensitive to many parasitic diseases. For example, trichostrongylosis, nematodiosis, etc. These diseases cause health problems and animal die prematurely and cause economic losses. Parasites cause many problems for wildlife, and although wildlife often adapted to the presence of parasite, but they do not adapt the adverse effect of parasites. In the wild, animals have a certain natural resistance or balance to the parasite, but in captivity, the changes in environmental and living condition affect the behavior and ecology. Hence, the susceptibility to parasitism is increased. The staff plays an important role in the transmission of parasite among animals in a zoo through their shoes, claws, food, hands, or with working tools. Another possibility of parasitic transmission is animals themselves when they are moved from one enclosure to another without proper parasitic treatment. The present study was to assess the helminths infestation in the white sheep at Kadwala Park, Faisalabad, and to collect and in vitro culture the eggs or oocysts from cases of helminths infestation where required, and materials and methods. A total of 120 fecal samples of wildlife sheep, Ovis orientalis, of all ages and sexes was collected in the period from October 2020 to March 2021 at the Wildlife Park, Gatwala, Faisalabad, from the ground after defecation at the morning of the day. About 50 to 100 grams of feces was placed in a polythene bag. The correctly labeled and properly numbered polythene bags containing the fecal samples with all required information was brought to the Laboratory Department of the Zoology, GC University, Faisalabad. All samples were examined and the samples were processed for microscopic examination. Adapting the special modification of the McMaster techniques for helminths, samples of 4.5 gram of fecal feces was weighed and mixed thoroughly in 40.5 ml of saturated salt, that is a sodium chloride solution, has the specific gravity of 1.20. Then stained using a sieve with an aperture width of approximately 1 millimeter and the debris discarded. Then 0.5 ml of the resultant solute was transferred into a double chamber McMaster slide. The slide was then left for 5 minutes to allow all the fecal samples, fecal debris with a specific gravity higher than 1.20 to sink the helminth's egg. EPG, that is egg per gram, number of is calculated by number of eggs multiplied by 100, where 100 is the dilution factor. Many nematode eggs are alike, and species such as Homunculus, Ostrategia, Trichostrongylus, Copria cannot be clearly differentiated from the eggs in fecal samples. Then the feces which are found positive for the egg parasite for identification, so exact identification, the using spatula and many feces were placed in a glass jar on petri dish which were closed and incubated at 27 degrees Celsius for 7 days. After incubation, samples were examined for larvae. Larvae were identified with the help of keys. Then shows these are the results. 4 types of nematodes or species were identified. These figures shows the eggs of the different samples of nematodes which were identified. The Trichostrongylus species egg. This is the Ostrategia species egg, and this is the Trichura egg. This table shows the overall prevalence of gastrointestinal wild nematodes among the wild sheep at Katwada Park, Faisalabad. 4 types of parasites were identified. That is Trichostrongylus species, Homunculus contutus, Ostrategia species, and Trichura. These table shows the occurrence of rate of prevalence of different species of nematodes. Out of 120 samples which were infected, samples were infected with the Trichostrongylus species, which was 58.33%, while the other Homunculus, Ostrategia or Trichurus 15%, 10% or 8.3%. The highest infection was with the nematode of Trichostrongylus species, having egg per range 100 to 200, mean value 157.92. And then others, while the egg per range was 100 to 300 are noted. This table shows the prevalence of gastrointestinal nematodes in male and females of wild sheep, so that is Ovis orientalis at Katwala Park, Faisalabad. The rate of prevalence in female species sheeps were higher as compared to male, which were 33% among female, while the 25% in male of the Trichostrongylus species. while for other species, Homunculus, Trichurus, or Strategia species also show the same ratio. This table shows the prevalence of mixed infection of gastrointestinal nematodes in wild sheep. Different mix of different nematodes infection were noted, and we observed the samples have mixed infections. The overall mixed infection prevalence was 8.32%. The prevalence rate vary from 4 to 1.6%, while the egg par range was 100 to 300, 250, 250. This table shows the seasonal fluctuations, seasonal prevalence of gastrointestinal nematodes in wild sheep, Ovis orientalis. Seasonal fluctuation had significant effect of gastrointestinal nematodes in wild sheep. A relatively high infection of nematodes was seen during the summer, which was 53.32%, while in winter, 38.32%. So the infections were higher during the summer season. Then the conclusion, samples were found infected with the gastrointestinal nematodes and overall prevalence were Trichostrongylus species, that is 58.33%, then Haemonchus contutus 15%, Trichurus trichura 8.33%, and Ostrategia species 10%. The infection among females, which was 65%, was higher as compared to male. Seasonal prevalence of gastrointestinal nematodes in summer and winter was 53.32% and 38.32% respectively. The nematode infection was higher in summer season as compared to winter. The range of egg per gram vary from 100 to 300 among the nematodes. The mixed infection, Trichostrongylus species and Haemonchus contutus, Trichostrongylus species and Trichura trichura, that is 2.5% and 1.6% respectively. The most prevalent nematode was Trichostrongylus species among wild sheep. These are the references which were cited. Thank you for your attention.
Thank you, Dr. Azhar Rafiq, for informative presentation. Our next presenter is Dr. Nosheen Aslam from Government College University, Faisalabad. She is going to present her work on evaluation of phytochemical analysis, free radical scavenging, and antibacterial activity of Carthamus oxyacantha. Please, Dr. Nosheen Aslam.
Bismillah. Assalamu alaikum. First of all, I would like to thank the Applied Zoological Society of Pakistan for providing me to present my paper in the 5th International Conference on Applied Zoology 2022. I am Nosheen Aslam from GCUS, where I am working as an associate professor in the Department of Biochemistry. The topic of my presentation is evaluation of phytochemical analysis, free radical scavenging, and antibacterial activity of Carthamus oxyacantha. Carthamus oxyacantha is known as highly valued medicinal plant. Carthamus genus belongs to family Astraceae, consisting 25 species. Previous studies indicated that various Carthamus species have been applied to cure a lot of diseases and illness like fever, cough, throat disorders, typhoid, swelling, antioxidant, cardiovascular problems, menstrual disorders, anticoagulant, anti-inflammatory, and calcium antagonist, etc. It is also useful in folk medication for a sedative and herbal medicine for anti-cancer, Different parts of Cathmus oxyacantha are rich in important bioactive compounds, phenolic, proteins, fatty acid, and phytochemicals. The seeds have purgative features or oil is important into therapy of scabs. It is also useful to lower pain, inflammation, enhances circulation of blood, and also possesses anti-hyperlipidemic characteristics. The phenols and flavonoids have potential free radical scavenging activity. These are useful therapeutically in curing many neurological diseases. These are major anti-cancer, anti-tumor, anti-mutagenic, and cardiovascular defensive mediators because of this capacity. Reported phytochemicals compounds are flavonoids, lutein and quercetin, tannins, triterpenes, and glycosides. The objective of this project is to find out the phytochemicals of extracts of Cathmus oxyacantha by preliminary phytochemical screening and to estimate antioxidant and to determine antibacterial activity of Cathmus oxyacantha extract. This is the herbarium specimen of Cathmus oxyacantha. First, the Cathmus oxyacantha aerial parts were collected in the month of October 2021 from the Cholistan desert of Tehsil Yazman, which is in the Bahawalpur of Punjab province, Pakistan. The collected parts of selected plant was washed thoroughly by using tap water for 2 to 3 times to remove any unwanted material. Clean air parts were kept for drying at room temperature. They were shed dried nearly 1 to 2 weeks. The dried aerial parts were grinded into a fine powder form by using grinder machine. The dried powder was kept in airtight jar and stored in dry and cool place to analyze. First of all, the extraction of plant was carried according to the standard procedure of maceration. And then the nearly 50 milligram of powder was soaked in methanol, ethanol, and ethyl acetate overnight, and the further procedure was adopted for the extraction of the selected plant. The preliminary phytochemical screening was done following the standard method described by these authors. The preliminary phytochemical screening tests are used to check the presence or absence of alkaloids, flavonoids, phenols, glycosides, saponins, tannins, terpenoids, steroids, coumarin, and quinones. And the isolation of possible pure phenolic components from methanolic extract of Cathamus toxicantha, which we named sample A, and ethanolic extract of Cathamus toxicantha, to whom the name we sample B of Cathamus toxicantha was executed by high performance liquid chromatography. The high performance liquid chromatography of methanolic and ethanolic extract was done in accordance with HPLC system of Promera to estimate the different phenolics and flavonoids. The stable free radical DPH was used for the estimation of antioxidant activity, and FCR was significantly used to determine the total phenolic content as described by Singleton in 1965 with modifications. The Escherichia coli and Staphylococcus aureus were tested in antibacterial activity. For this purpose, we use disc diffusion assay, and gentamicin was used as a standard, and zone of inhibition was studied as comparative method to measure effectiveness of Cathamus of the cantha extract as an antibacterial agent. Here are the results. Alkalides and glycosides were present abundantly in ethanolic and methanolic and ethyl acetate extracts. Flavonoids were present in methanolic and ethanolic extracts. Flavonoids were not detected in the ethyl acetate extract. Similarly, saponins were detected in good quantity into the ethanolic extract but was absent in methanolic and ethyl acetate extract. Other phytochemicals, phenols, terpenoids, steroids, quinones, and phenols were present abundantly in the methanolic and ethanolic extract, but they were deprived of ethyl acetate extract. These results indicates that DPPH free radical scavenging activity of ethanolic extract is greater than methanolic extract of Carthamus oxyacantha, which indicated strong antioxidant activity. The DPPH radical scavenging values of both methanolic and ethanolic extract of selected plant was greater than standard, which is 76.38, indicating strong antioxidant activity of plant. The percentage DPPH scavenging value of extract was increasing with increasing concentration. It was resulted that methanol extract of Cathamus oxyacantha have high percentage of total phenolic content rather than the ethanolic extract. The percentage value of total phenolic content was determined from the standard Calvin equation of gallic acid, which is used as standard. The result indicated that methanolic and ethanolic extracts of plant contains greater number of phenolic compounds than ethanolic extract. Although both extracts of plant, ethanolic and methanol, showed strong antioxidant activity, the antibacterial activity of ethanol, methanol, and ethyl acetate extracts were separately tested against the studied bacteria. Ciprofloxacin was used as positive control to determine the sensitivity of bacterial strain. The antibacterial activity of Cathamus oxyacantha aqueous extract of were screened against selected human pathogens, Staphylococcus aureus and E. coli. Lower molar concentration could not display inhibitory effect against Staphylococcus aureus, while 200 micrograms showed maximum zone of inhibition against test organism. Similarly, in the methanolic extract, same, the lower concentration could not display inhibitory effect against E. coli, while 200 micrograms showed maximum zone of inhibition against test organisms. It is the HPLC chromatogram which revealed the presence of polyphenols like catechin, fumaric acid, gallic acid, chlorogenic acid, cyanic acid, caffeic acid, ferulic acid, and quercetin with retention time. When methanolic extracts were subjected to HPLC column and the obtained record were superimposed on the retention time and values of this extract. HPLC HPLC-based comparison showed that there is a variation with regard to retention time within different extract. As you can see in there is a different concentration. Again, we can say the presence of different phytochemicals within different extracts, indicating that the presence of different phytochemicals in the methanolic and ethanolic extract. So, by this test, we can conclude the different parts of plants contain many phytochemicals like stain and catechin, which perform its activity as a natural oxidants, flavonoids, and phenols. Phenolics are involved in the chelation of metal ion, which can catalyze the formation of frost and promote lipid peroxidases. Total phenolic contents and total reducing capability assay confirm the extracts have active antioxidants. The results reveal that the active components of the plant inhibit growth of approximately all the tested microorganisms. These are the references. Thank you for your attention.
Thank you, Dr. Nosheen Aslam, for nice talk. Let's move toward our next presenter, that is Dr. Abdul Qayyum from University of Education, Lahore. His topic of presentation is estimation of selected metals in the blood of prostate cancer patients and reference. Please, Dr. Abdul Qayyum.
Bismillahirrahmanirrahim. First of all, congratulations to ICAZ-2022 team for organizing such a good event of 5th International Conference on Applied Zoology, abbreviated as ICAZ 2022. The topic of my presentation is estimation of selected metals in the blood of prostate cancer patients and reference. My name is Dr. Hafiz M. Abdul Qayyum. I am Assistant Professor in the Department of Chemistry, University of Education, Lahore, campus, Faisalabad. First of all, cancer. Cancer is not a single but group of diseases characterized by uncontrolled growth of cells. Cancer develops only in cells with damaged genes. It is a multifactorial and multi-mechanistic disease which can spread in any part of the body. Stepwise development of cancer involves different stages such as initiation, promotion, progression, and metastasis. Prostate cancer. Prostate cancer is one of the most common cancers in men. Screening for and management of early prostate cancer is one of the most challenging and controversial issues in all of the medicine. Risk factors. There are many risk factors involved to spread the prostate carcinoma. Number one is non-modifiable factors. Paramount among these is age. In unscreened population, prostate cancer has the steepest age incidence curve of all cancer and increases at approximately the sixth power of the age. Only 25% of the cancer are diagnosed before the age of 65 years. Genetic factors. The relative risk of developing prostate cancer is higher in men who have a first-degree relative with prostate cancer. This risk is higher in men under 65 years age. External exposure. Both ionizing radiation and ultraviolet radiation from the sun exposure have been linked to prostate cancer. Exposure to metals, including toxic metals such as aluminum, nickel, cadmium, mercury, and lead, have a major role in the development and pathogenesis of prostate carcinoma. In damaged DNA, three processes occur. Number one, gene mutation. Number two, inactivation of tumor suppressant gene. And number third is the activation of mutated gene. Due to above three processes, normal body regulation mechanism fail, and then coronal expansion and additional mutation takes place in the cells. Significance of selected metals. Essential metals are required for normal growth and development in human body, while their deficiency led to various ailments. Therefore, metals play both curative and preventive role in combating diseases. Trace metals are required at optimum concentration for proper functioning of the human biological system. Risk factors. There are many risk factors involved to spread the prostate carcinoma. Number one is non-modifiable factors. Paramount among these is age. In unscreened population, cadmium is non-essential for humans. It induces nephrotoxicity, immunotoxicity, and osteotoxicity. IARC classified cadmium as a group 1 human carcinogen, and cadmium develops cancer. External exposure. Both ionizing radiation and ultraviolet radiation from the sun exposure have been linked to prostate cancer. Exposure to metals, including toxic metals such as aluminum, nickel, cadmium, mercury, and lead, have. Copper occurs in more than 300 enzymes. Its toxicity may lead to prostate cancer or cancer and ovarian cancer, but its deficiency may cause anemia, diabetes, and osteoporosis in children. Iron is mostly utilized in hemoglobin. However, its toxicity typically damages cells in the heart, Parkinson's disease, and develop liver, thyroid, lung, and stomach cancer as well. Mercury and its compounds are extremely toxic and must be handled with care. The most toxic form of mercury are organic compounds such as dimethylmercury and methylmercury. Mercury can cause both chronic and acute poisoning. Following are the major objectives of the study: to obtain a baseline data for selected essential and toxic elemental concentration in blood of prostate carcinoma patients with respect to healthy subjects, to investigate the mutual relationships among the essential and toxic elements in terms of correlation coefficients, to find out the viable correlation among the essential and toxic elemental levels in the blood of prostate carcinoma patients and controls, to find out the significant difference in concentrations of essential and toxic elements in various types and stages of prostate carcinoma in patients. Experimental methodology consists of following 4 steps: sample collection, sample preparation, meta-analysis, and the last one is the statistical analysis. Sample collection. The blood samples were collected on a volunteer basis from the newly diagnosed cancer patients admitted in Punjab Institute of Nuclear Medicine, Faisalabad. Samples from healthy subjects were also collected on a volunteer basis. Information regarding donors' diet, age, abort, smoking habits, health status, occupation, cancer type, and stage was also recorded on a proforma. Blood samples were collected through veiny pincher method from the antecubital vein by using appropriate precautions to prevent exogenous contamination. Sample preparation or processing. The reaction was carried out on hot plate. 10 ml of nitric acid was added in a precisely weighed sample and then left for 5 minutes. After this, 10 ml of perchloric acid was also added and heating on hot plate at 80 degrees Celsius for 4 to 5 hours. Evolution of white dust fumes showed completion of digestion process. Final volume was adjusted with doubly distilled water. Statistical treatment of data involve minimum, maximum, mean, median, standard deviation, standard error, skewness, correlation, kurtosis, and quartile distribution is involved. Now the finally results and discussion. Statistical distribution for the concentration in microgram per gram of selected metals in the blood of prostate cancer patient as shown in this table. On the average basis, average concentration of iron, magnesium, and cobalt were higher in the blood sample of prostate cancer patients. Similarly, statistical distribution of concentration of selected metals in the blood of healthy subjects as shown in this table. Average concentration of iron, magnesium and cobalt were higher in the blood of healthy subjects. However, on comparative basis, average concentration of iron, magnesium, cobalt, chromium and cadmium were higher in the blood of prostate cancer patients than the healthy subjects. Correlation coefficient matrix for selected metals in the blood sample of prostate cancer patient as shown in this table. This table shows that essential and toxic metals were correlated in the blood samples of prostate cancer patients. Likewise, correlation coefficient matrix for selected metals in the blood sample of healthy subjects as shown in this table. In this table, essential metals were also correlated with each other instead of toxic metals as shown in this table. Lastly, we discuss comparative evaluation based on demographic characteristics. Comparative average concentration of selected metals in the blood of prostate cancer patient and healthy subjects as shown in this figure. On the average basis, average concentration of cadmium, chromium, tin, mercury were highest in the blood of prostate cancer patient than healthy subjects. Comparative average concentration of selected metals in the blood of vegetarian and non-vegetarian patients and healthy subjects as shown in this figure. Average concentration of cadmium and mercury was highest in non-vegetarian patients as compared to non-vegetarian reference, whereas magnesium and cadmium was highest in vegetarian patients as compared to vegetarian controls. Comparative average concentration of selected metals in the blood of prostate cancer patients and healthy subjects based on smoking and non-smoking habits as shown in this figure. This figure shows that average concentration of cadmium, chromium and iron was highest in smoker patients than smoker controls. Similarly, mean levels of copper and iron was highest in non-smoker patients as compared to non-smoker controls. Levels of selected elements in the blood based on stages 1, 2, 3 and stage 4 of prostate cancer patient as shown in this figure. At stage 1, cadmium and cobalt were highest, whereas stage 2, nickel was highest in the blood of prostate cancer patients. Similarly, at stage 3, nickel was elevated, and stage 4, chromium, mercury and magnesium was highest in the blood of prostate cancer patients based on stages. Levels of selected elements in the blood based on types of prostate cancer patient as shown in this figure. In squamous cell carcinoma, magnesium, cobalt and chromium was highest as compared to other types of prostate cancer patients. In transitional cell carcinoma patients, iron and nickel were highest or elevated, whereas small cell carcinoma patients have cadmium, mercury and copper was highest in the blood of prostate cancer patients based on types of prostate carcinoma. Finally, conclusions. The average concentration of cobalt and cadmium were appreciably higher in the blood of prostate carcinoma patients than controls. Similarly, average levels of chromium and cadmium were significantly elevated in the blood of urban patients than urban controls. The selected metals like mercury and cadmium were maximum in the blood of non-vegetarian patients than non-vegetarian controls. In comparison of smoking and non-smoking habits, relatively higher levels of chromium and cadmium were elevated in the blood of patients with smoking habits, and magnesium and iron were maximum in the smoking controls. The correlation study revealed noticeably different mutual variations of metals in the blood of two donor groups, which evidences that the prostate carcinoma conditions were significantly affecting the metal imbalances in humans as well. Recommendations of the present study. The present study pertaining to select essential toxic metals linked with prostate patients of prostate carcinoma. Various sessions are described below. Large-size clinical research should be conducted, especially considering long-term exposure of essential toxic metals. More randomized study should be done with large elements. Various biological specimens such as scalp hair, saliva, nails, and milk should be used for diagnosis of this malignancy. Thank you.
Thank you, Dr. Abdul Qayyum, for delightful presentation. Let's move to the next presenter, that is Dr. Shahid Shah from Government College University, Faisalabad. His topic is knowledge, attitude, and practices of pharmacogenomics among senior pharmacy students, a cross-sectional study from Punjab, Pakistan. Please, Dr. Shahid Shah.
Good morning. I am Dr. Shahid Shah from the Department of Pharmacy Practice, Government College University, Pakistan. First of all, I would like to say thanks to Applied Zoological Society of Pakistan for giving me the opportunity to share my research work on this platform. The topic of my research is knowledge, attitude, and practices of pharmacogenomics among senior pharmacy students, a cross-section study from Punjab, Pakistan. So pharmacogenomics is a key to the success of personalized medicines as it minimizes inter-individual fluctuations in drug response. In developed countries like USA, pharmacogenomics training programs are carried out regularly. So we can see the significant improvement in pharmacotherapeutic outcomes. But in developed countries like Pakistan, such practices are very rare. So our objective was to assess the knowledge, attitude, and practice of senior pharmacists toward pharmacogenomics in Punjab, Pakistan, because pharmacist is considered the pillar of health care system architectures. So questionnaire-based cross-sectional study was conducted by administering the questionnaires to the senior pharmacy student from different public sector universities like Government College University, Faisalabad, Mohammed University Multan, Islami University Bahawalpur, Punjab University Lahore, and so many other universities. So all the participants gave informed verbal consent rather than the written consents because there is no clinical interventions in our study. So questionnaire was developed after literature survey, and the main points of the questionnaires were demographics, educational exposure to pharmacogenomics, knowledge of pharmacogenomics, knowledge of FDA recommendations about pharmacogenomics, testing of drugs, attitude toward pharmacogenomics, and the practice of pharmacogenomics testings. So to validate our questionnaires, our survey instrument was reviewed by the investigators for content, clarity, relevance, and ease of understanding of the questions. And we also used the pilot test for the questionnaires. The sample that we included in pilot testing was not included in our final results. And the Cronbach's alpha values for knowledge, attitudes, and the practice was found 0.78, 0.69, and 0.71 respectively, that indicate the good internal consistency. So data was collected by preparing the questionnaires and distributing through hard copies to all the senior pharmacists, and the participants or the senior pharmacists were told that they could withdraw from the study if they wanted. So our survey response rate was 78%, and we excluded the duplicate response. So here are the dependent variables. For example, first one is the educational exposure to pharmacogenomics. And this was assessed using 5 questions. And the next is the student's knowledge of pharmacogenomics, that was determined using 10 questions. And each questions had to be answered like I know means 1 score, I don't know, we gave 2 scores, and I'm not sure, we gave 3 scores. So knowledge score less than 14 was considered good knowledge. And similarly, knowledge of FDA recommendations for pharmacogenomic testing of drugs was determined using 6 questions about the labeling of different drugs as evidenced by the FDA. So attitudes of students were assessed using 4 questions, and each was answered like yes, 1 score, I'm not sure, 2 score, and no means 3 scores. So attitude score of less than 6 was considered the good attitudes of the senior pharmacist students. And the last one, dependent variables, was the practice of pharmacogenomic testings that include 5 questions and each to be answered with always, at least once in a month, at least once in a year or semester, and at least once in a program or never. So practice score in the range of 5 to 8 was considered good practice. So independent variables that we use were the demographics that include the age, genders, and the study institutions. So here are our results, and here you can see the majority of the students or the senior pharmacists were having the age of 20 to 25 years. And here are the different universities from which we collected the data. So here, the educational exposure to pharmacogenomics, you can see that most of the students, 93.2%, studied about the inter-ethnic and inter-individual variations in drug response during their university educations. And most of the students learn about pharmacogenomics from the university lectures. And usually 3 to 5 lectures they attended from the department to describe the effect of various genetic variants on the drug response. 52.1% of the students have been able to comprehend the implications of diverse genetic variations on drug response, and 60.3% of the students clearly describe the effects of genetic variants on drug response. Here is the basic knowledge of pharmacogenomics, and from this table we concluded that the mean knowledge score that was 12.6, and as I already told you, that 12.6 range under the good considerations. So other one is the scatter plot matrix and a linear regression model. So here is the gender-wise distributions, and we found here that female had higher knowledge than males, and the good knowledge positively and significantly affected the knowledge of pharmacogenomic testing and students' attitudes. Here you can see the gauge chart that shows us the mean score of knowledge of pharmacogenomic testings, attitude, and the practice. And the main score of knowledge and attitude is good as compared to knowledge of pharmacogenomic testing and practices. So here is the knowledge about the specific validated pharmacogenomic testings. So here is the attitudes towards pharmacogenomics, and this graph shows the gender-wise and the age-wise variations of attitude and practice for knowledge, and the linear line, straight line shows the linear regressions for both male and female. So female, we found that female had 2 times more likely to have good attitudes compared to males, and the good attitude positively and significantly affected the knowledge of students. Here is the practice of pharmacogenomic testings, and we use multivariate logistic regression models. And from these tables, we concluded that female have more practices as compared to male for the pharmacogenomic testings. So our conclusion is that senior pharmacy students in Punjab, Pakistan had good knowledge and attitudes towards pharmacogenomics. However, the practice in this regard is not up to the mark. So there is a gap in practice students or the pharmacists that must be fulfilled to improve drug therapy and to optimize drug response. Thank you for your attention.
Thank you, Dr. Shahisha, for productive talk. Our last presenter of this session is Dr. Amara Tahreem, Government College, University, Faisalabad. Her topic of presentation is anti-hyperglycemic potential of 2-E-3-phenyl 1,3-pyridinol 2-propanol in alexionized hyperglycemic rats. Please, Dr. Amara Tahreem.
Bismillahirrahmanirrahim. My name is Amara Tahreem. I'm a PhD student in GC University, Faisalabad. First of all, I would like to thank ICAZ-2022 for giving me the opportunity to present my research work. So the title of my research is Anti-Hyperglycemic Potential of 2-E-3-phenyl 1,3-pyridinol 2-propanol in allogenized hyperglycemic rats. As my research is related to diabetes, we know that diabetes is a life-threatening disorder due to defects in insulin secretion, action, or both. Insulin is a hormone which is produced by the pancreas. So there are two types, main types of diabetes: type 1 diabetes and type 2 diabetes. In type 1 diabetes, the pancreas does not produce insulin. However, in the case of type 2 diabetes, enough insulin is not produced by pancreas, or in some cases, the insulin produced inside the body does not work properly. However, in case of gestational diabetes, it is mainly developed mostly in the pregnant ladies and disappeared later on. These are the symptoms of type 1 diabetes. Pancreas does not produce insulin in type 1. So these are the symptoms: frequent urination, unexplained weight loss, fatigue, weakness, blurry vision, irritability, and mood swings. However, in the case of type 2 diabetes, along with these symptoms, some other factors also contribute, that are obesity, smoking, hypertension, and other risk factors are fast food and sedentary lifestyle. And that may lead to further diabetic complications. Role of oxidative stress. Oxidative stress is a very threatening problem in many disorders. Hyperglycemia is developed due to the auto-oxidation of glucose, or due to the insulin resistance inside the body. So as a result of hyperglycemia and insulin resistance, the reactive oxygen species inside the body become overreactive, or the overproduction of reactive oxygen species may lead to oxidative stress. Both these factors may contribute to beta cell dysfunctioning, and beta cell dysfunctioning in return also cause the hyperglycemia and insulin resistance. So these are all are interrelated factors that may contribute to diabetic abnormalities. 2-E-3-phenyl 1,3-pyridinol 2-propanol is my research compound. This is basically a chalcone derivative. So what are chalcones? Chalcones are precursors of flavonoids and isoflavonoids, and we know that flavonoids are very important polyphenolic compounds, which are obtained both from edible plants and can be synthesized also. So these are the flavonoids are present in most leguminaceae family, or show very important biological activities like antioxidant properties, anti-inflammatory properties, anti-cancer properties, and neuroprotective effects. So what is the objective and need of the project? The objective of this study was to determine the anti-hyperglycemic activity of this research compound, and to determine the antioxidant activity of the compound. These are all the therapeutic targets of chalcones, which have been evaluated in previous studies for their anti-diabetic potential. For example, the chalcones might cause the inhibitors of these therapeutic targets inside the body. As a result of this inhibition of these enzymes, diabetes can be cured. So methodology, first of all, we took 50 healthy albino Winster rats from Annual House of the Institute of Pharmacy, Physiology and Pharmacology, University of Agriculture, Faisalabad, and then kept them for their acclimatization period. After acclimatization period, we divided them into 5 groups, each containing 10 rats. So for negative control group, we have given only the routine diet. For positive control group, we injected the Lexon monohydrate intraperitonially, and same was the case with the other 3 groups. So diabetes was induced, and the diabetic levels were checked regularly before the start of the treatment until the rats showed the levels of 300 milligram per deciliter. So we ensured that they are not diabetic, and further treatment is can be started. Then we started treating the group 3 with the bencalamide, that is a standard antidiabetic drug, and group 4 and group 5 with chalcone derivative 5 ml per kg orally, and treated to group with chalcone derivative 10 ml per kg orally. So timeline was about for 28 days. So during this treatment period, we did the monitoring for physical parameters like body weight, feedback, and water intake. Levels were checked on weekly basis. While chemical analysis, fasting glucose levels were also checked on a weekly basis. Weight and fasting glucose levels were checked, and the results showed that treatment groups restored the weight. So weight was reduced in positive control group that might be linked to the glucose auto-oxidation and protein breakdown due to the insulin diabetes induction. But after the treatment with the both treated 1 and treated 2 groups, we have seen that they restored their weight. Same was the case with fasting glucose levels, that these levels were high in positive control group and the other 3 groups, but after treatment with glibenclamide and treatment 1 and treated 2 groups for 0, 14 and 28 days, we can see that the treatment groups, in treatment groups, the fasting glucose levels were markedly reduced. That might be due to the anti-hyperglycemic potential of the chalcone derivative. In feed intake and water intake, the levels of feed and water intake were gradually increased in after the after the diabetes induction, but restored were increased in after diabetic induction, but were decreased gradually after after giving the treatment 1 glibenclamide treatment and the chalcone derivative both treatments. These groups restored the water intake and feed intake. So after 4 weeks, we did the decapitation, and blood samples were collected that were kept in the clock activator tubes, and then after centrifugation, serum samples were collected, and then tag, toss, glucose, insulin, RFTs, and cholesterol levels were measured. Tissue samples were collected, and pancreas was kept in the formalin, and then in formalin for histopathological examination, and then their slides were made, and then micro photographs were analyzed later on. So these are the results of serum insulin levels. We can see that treatment group showed decreased serum glucose levels as significantly as compared to the positive control group. That might be linked to the, again, with the anti-hyperglycemic potential of chalcone derivatives. Same is the case with the serum insulin levels, that the serum insulin levels were increased in the positive control group, were decreased in the positive insulin positive treatment group, control group. However, in glibenclamide treated and treatment 1 and treatment 2, we can see that insulin levels were increased. That might be linked to the regenerative capacity of the chalcone derivative on the pancreatic beta cells. In the case of serum toss and tag, total oxidant status of the positive control group was markedly increased as compared to the glibenclamide and treated 1 and treated 2 groups. Same is the case, and in the case of total antioxidant capacity levels, we can see that total tag levels of treated 1 and treated 2 and glibenclamide are significantly increased as compared to the other glibenclamide treated group and positive control group. This increase in the total antioxidant capacity levels of these 2 groups might be linked to the reduction in the insulin resistance inside the body after treatment. So serum creatine and urea levels, these are the breakdown products of protein, and creatine are obtained from the muscles. We know that as a result of overproduction of reactive oxygen species, oxidative stress happened during diabetes that may cause the lipid peroxidation. Also, due to the increased absorption of alloxan in the kidney tubules, so that might affect the creatinine levels and urea levels and their excretion also. So in diabetic groups, we can see that the level of serum creatinine and urea were increased. However, after treatment, we can see that in treatment groups, these levels are markedly reduced. Serum total cholesterol and triglyceride levels, these levels are increased due to the inactivation, might be due to the inactivation of lipoprotein enzymes. However, after treatment, we can see that these levels are markedly restored or reduced. Might be it might be linked towards the regulation of lipoprotein enzymes. So this is the histopathology of pancreas. We can see in the tissue that the cells, parenchyma cells, are in very refined form, and the cellular contents are present. However, in the case of positive control group, we can see that the cell, there is a necrosis or might be edema, and there are spaces among the cells are increased, and their destruction of pancreatic beta cells can be seen easily. However, in the case of glibenclamide treated group, there is a tendency towards recovery. Same is the case with treated one group. We can see that the edema is reduced and necrosis is or swelling is this spaces among the cells is markedly reduced. And this is the complete recovery we can see after treatment with chalcone derivative. This might be related to the regenerative and anti-hyperglycemic activity of the research compound. So on the basis of this study can be concluded that 2-ethylphenyl 1,3-pyridinyl 2-propane 1 contains significant antioxidant capacity and anti-hyperglycemic potential. This compound should be evaluated critically at all clinical stages to ensure its therapeutic and toxicological profile to meet the demands of diabetics. Thank you.
Thank you, Amara Tahreem, for informative presentation. Our next moderator will be Sabariyaz from Government College University, Faisalabad. Thank you for being attentive. Stay tuned.